So now that we have added the Benedict solution into the different test tubes, the next step is to take these test tubes and add them in the boiling water.
That's right.
In a food, carbohydrates may be present in the form of a simple sugar, such as glucose, or named as dextrose, and fructose.
Foods also contain a double sugar. Two simple sugars combine together to form a double sugar. For example, sucrose and what did I say? Oh yeah, I forgot it almost. Sucrose in sugarcane and lactose in milk.
In many cases, foods contain many simple sugars combined together. For example, starch and cellulose in plants and glycogen in animals.
Well, I think there are names for these types of sugars. Simple sugars are called monosaccharides.
Double sugars are called disaccharides.
And many simple sugars combined together are called polysaccharides.
Okay, well, let's now take only carbohydrates. We take a simple sugar such as glucose and fructose. We also take a double sugar such as sucrose and a polysaccharide such as starch.
Okay, for this experiment, we need 5 test tubes. As she said, glucose powder, fructose powder, sucrose and soluble starch, and also an beaker filled with water and an electric stove.
So the first step in this experiment is to take the beaker filled with water and put it on the stove and bring it to the boil. In the meantime, we take 6 separate test tubes and put into them tap water to about a depth of 2 centimeters.
Okay, but then we have to leave one test tube to be of control and label it as control. To each of the other test tubes, we respectively add the different carbohydrates we have here and dissolve them in the water. Then after, we label them respectively.
Hannah, could you please bring us some distilled water so that we can add a few into the test tubes?
Sure. Here you go, Kevron.
Thank you very much, Hannah. So now, like I said before, I'm going to add distilled water, into 6 test tubes to a depth of about 2 centimeters. So can one of you please help me?
Okay.
Here are the pipettes.
Thank you. There you go.
Okay. But now we have to leave one test tube to be a control.
Oh, yes.
So I will add some water into this one. Okay. Then after, I will label this as control. Thank you. So now I think we must add these carbohydrates into their respective test tubes, which we have labeled. I will start by adding the glucose.
Hannah?
Okay. We have here sucrose.
I'll add in the starch.
Okay. Well, I should probably add the fructose then.
Should we mix them first?
Yeah, I think
Okay,
so.
I think we should.
We now add a drop of Benedict's reagent to each tubes until it's blue. Donna, can you please help me?
Okay, sure. I think you should add some more. Okay, Ron, do you mind mixing this?
No problem.
Here's the sucrose.
You can do the rest of
That's
this.
Oh, I don't mind.
Okay.
And lastly, here's the starch.
So now that we have added the Benedict solution into the different test tubes, the next step is to take these test tubes and add them in the boiling water.
That's right.
Can you guys help me?
Okay, I'll take the test tube round. Okay, well, I will start by adding the glucose first.
Be careful.
Hannah, would you do the rest?
Okay. I have sucrose here. And here's fructose. And lastly, the starch.
But don't forget the control.
Oh yes.
Now, we'll have to leave them there for a few minutes and see if there's any color change.
As you can see, the colors change from blue to orange in tubes containing and glucose. There is no color change in the tube containing the control. The soluble starch and also sucrose.
I will bring back the test tube rack.
But I noticed that the color of the water in the control is not blue. I think we forgot to put Benedict's solution into it in the first place.
Oh yes.
So we should do the experiment for the control only, don't you think?
Okay, that's a good idea.
Let's put some drops of Benedict's solution into the water. A little more, please.
Okay, well that should do.
Yes. So now I'll put this in the boiling water and we can see if there's a change in color just after a few minutes.
Okay.
But the other experiment seems to have worked.
Yeah. Oh well,
So-
it does actually.
Yeah. So what kind of conclusion can we make after this?
Well, we know that all 4 of these are carbohydrates. We have to see what is common for glucose and fructose. How are they different from sucrose and starch?
Glucose and fructose are monosaccharides. By the side, sucrose is a disaccharide and starch a polysaccharide.
Well, so I think we can conclude and say that monosaccharides change the color of Benedict's solution into, which has a blue color, from the blue color it has to orange, just like glucose and fructose did.
Oh yes. But could you please explain to me why there is a change in color?
I have read in our textbook that the Benedict solutions contained copper 2, and monosaccharides reduce copper 2, which is blue, to copper 1, which is orange. Monosaccharides give electron to copper 2. That's why monosaccharides are called reducing sugars.
Thank you.
So now let's check the control and see if there's any kind of color change. Thank you, Miss. So as you can see, the water in the control is still blue. There is no color change.
Oh yes.
That's right.
So I think we have had a successful Benedict's test.
We have indeed.
Yes.
But we mustn't forget that monosaccharides, like glucose and fructose, dissolve faster in water than cane sugar. They are also sweeter than cane sugar.
Yeah, you're right.
Yes.